our copy and paste mechanism. So in this case here, this is my transposon, this little piece of DNA. A copy of it was made, okay, and it's been inserted somewhere else along the chromosome. And so it has, quote unquote, jumped to a new location. You can also have what's called a retrotransposon. And a retrotransposon is an RNA intermediate is made, and then that reverse transcriptase is used to make the new DNA segment that is then inserted somewhere else on the genome. So instead of making a DNA copy, like what happened here, transcription would actually happen, and this would be an RNA. For a retrotransposon, it would take this DNA into RNA, and then the reverse transcriptase would transcribe it back into DNA, and so then it can be inserted somewhere else along the genome. So to just sum up these types real quick, you've got your transformation again. Oops, sorry about that. Okay, so you've got your transformation. There's your foreign DNA that was in the environment that gets taken into the cell. Okay, your transduction, where the virus is going to inject pieces of DNA, possibly from another bacterium, and then the conjugation. You can see the sex pill is there, the mating bridge, and so it's sharing DNA between the two. Okay, so now that we've seen some of the ways that bacteria can get variation, we're going to look at how we can use some of this in the form of biotechnology. And so biotechnology is just our manipulation of the natural processes so that we can serve societal needs. So basically we're tinkering with the genomes and the genes of organisms. And they can be done for a variety of reasons. If you look at this here, you can see that you can generate organisms, plants that are resistant to pesticides. You can, have pest resistant, not resistant to pesticides. You can have genes that are used to help convert bacteria into bacteria that can clean up toxic waste. Okay, for medical purposes, the production of human growth hormone, insulin, so a variety of reasons that we might use this. Okay, as we go through this unit, we'll also start to be bringing up, y'all will need to be thinking about the bioethics behind this. What is your line that you think that this is not okay and that we've gotten out of control and we need to stop what we're doing with that? So we've got 4 basic kinds of biotechnology that we're going to talk about. We're going to do 1 today, and then the other 3 will be on the next video. So the first one here is what's called transgenic. And with transgenic biotechnology, we're just talking about mixing genetic material from multiple species, across the species. So this would be mixing the bacterial, mixing the human DNA into the bacterial DNA so that the bacteria will produce, human insulin. That would be an example of transgenic. Another one would be reproductive cloning. And like I said, we're going to talk about each one of these separately, okay, and a little bit more in depth, like starting with reproductive cloning. That'll be on the other video. But reproductive cloning is exactly what it says. We're making exact clones or exact copies of the particular species. Okay, another would be reprogramming the cells. Okay, this will talk about, we'll talk some about stem cells, okay, and differentiation of the cells and trying to get them to differentiate into other cells. Okay, and then the last kind would be what's called forensic biotechnology. And with forensic biotechnology, this is what we would have talked about some your freshman year with gel electrophoresis, where you read the banding and determine who's the father, which criminal committed the crime. Okay, those would be examples of forensic biotechnology, using restriction enzymes, electrophoresis to distinguish people. So the rest of our video, we're just going to spend a few minutes focused, like I said, on the transgenic biotechnology, and we'll cover the rest later. We're going to walk through the transgenic biotechnology using an example. Remember again, the transgenic is when we are going across species. So we're mixing DNA from different species of organisms. And so we're going to use the example of human growth hormone. Okay, remember, human growth hormone is produced in the pituitary gland. Okay, and obviously human growth hormone has a big impact on our stature, okay, our growth and our development as a whole. Okay, and so the human growth hormone is a peptide hormone If y'all remember peptide hormones, that means it's a protein-based hormone. So that means it has to go through that cell signaling pathway. Remember, it has to go through reception, bind to the receptor on the membrane protein, and then the signal transduction. This is like the free response question you've seen before. And then it has an impact on the DNA. Remember, it can either turn genes on, turn transcription off, turn it off. Remember, that's different than a steroid hormone. Because remember, steroid hormone is lipid soluble. They're able to fit through the cell membrane and the nuclear membrane, and they just go straight in and act on the DNA. And so with human growth hormone, obviously if we're not producing enough, then growth is severely stunted. People are significantly smaller in stature. And so human growth hormone used to have to get human growth hormone, it had to be extracted from deceased humans. And so there obviously wasn't very much available. Okay? And so research came up with basically the idea of taking the gene that codes for the human growth hormone, okay, taking it out of the human genome, putting it into a plasmid, and making recombinant DNA. Okay, so we're taking the human growth hormone gene, okay, putting it into a plasmid. So now I get recombinant DNA. Remember, recombinant comes from 2 different sources. So now I have recombinant DNA, and now that recombinant DNA can hopefully be taken up through transformation. So remember the outside, that pickup of foreign DNA from the outside environment. So this new plasmid, this recombinant plasmid that's been made, okay, would be in the environment that the bacterial cells are in. And so hopefully the bacterial cells will take up this new DNA, and so every time they reproduce, they're going to be they're able to synthesize the human growth hormone, and every time they reproduce, their progeny, their offspring, also have this gene. Look a little closer about how they actually got the human growth hormone gene of interest, because the gene to produce the human growth hormone is just one small section of the chromosome. And so we are able to pull that gene out of the chromosome using what are called restriction enzymes. And so restriction enzymes look for particular sequences of DNA. They look for what's called a recognition sequence, and when they find that recognition sequence, they will cut the DNA in that spot. So for instance, in the picture up here, we've got the recognition sequence for this particular restriction enzyme is GGATCC. So every time it sees that sequence, it's going to cut the DNA there. And so this restriction enzyme has moved down, it's cut the DNA there. Okay, a second restriction enzyme has come down the other side, and so we've cut the DNA here in these various places, resulting in this DNA fragment. We essentially cut that DNA out of the genome, and it creates what are called sticky ends. And so these sticky ends, they are sticky because they are complementary to other strands that have been cut with the exact same restriction enzyme. You'll see this overhang here. Okay? Basically, you've got a strand of DNA. So when I've cut it here, I've cut this DNA in such a way that I have this overhang. Okay, and so when I have this overhang here, so I've got this area and this area, those are, like I said, those are what are called your sticky ends. And so you would have, so there's the rest of my double strand. Okay, I would have this area here that's able to complementary base pair with something else. And it's going to be able to complementary base pair with another gene that's been cut, or another piece of DNA that's been cut with the exact same restriction enzyme. And so we'll use the DNA ligase. Remember, the ligase brings them back together. Okay, so we use the DNA ligase to splice these sticky ends together. And so only DNA that's been cut with the exact same restriction enzyme can be spliced together, because like I said, they'll have matching sticky ends. So for instance, when you're done up here, let's clean this up a little bit so you can see it a little bit better. So when I'm done up here with this particular DNA fragment, I'm going to have hanging off of the end, and blah, 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 blah. Okay, hanging off of the end of the two sides, I'm going to have these overlap sections here. And these are, like I said, those are my sticky ends. Those are able to complementary base pair with something else that's been cut with the exact same restriction sequence. And then the ligase can come in and splice those together. So you can see that gene splicing happening here. Okay, so the restriction enzyme went in looking for the particular sequence. Okay, and so when it found that, that's called the restriction site, and it has cut the DNA there. Now I've got my sticky ends, and those sticky ends can be combined with other DNA that was cut with that same restriction enzyme, and the DNA ligase then seals those strands up now. You can see now here how researchers were able to use this method of cutting out the gene of interest and inserting it into the plasmid so that we can produce more of this human growth hormone. So here we've got again, the gene of interest over here, okay, and it's been cut with the restriction enzyme. So there's the piece of the gene of interest that's been cut out with the restriction enzyme, and the bacteria plasmid, okay, was cut with the exact same restriction enzyme. So they have the same sticky ends now. So now they are able to complementary base pair with one another, and the DNA ligase can seal them back together. So now we have this recombinant DNA, this DNA that's a mix of DNA from two different sources. And so by using the process of transformation, okay, so this foreign DNA is in the environment around this bacterial cell right here, okay, it takes in the recombinant DNA. So now all of these bacteria that are reproduced are going to contain that recombinant DNA. And so now when they transcribe and translate that plasmid, they're going to transcribe and translate that gene of interest, which codes for whatever particular, in this case, the human growth hormone that we're trying to produce a lot of. A couple other examples of this would be the production of insulin. Okay, insulin using either yeast or this is what's called a safflower plant. Okay, these can be used to produce human insulin. And remember, insulin is the peptide hormone that helps take up glucose out of the blood into the cells. People that are diabetic that may not produce insulin, they may need synthetic insulin. And so the yeast and the safflower plant can be used to produce the insulin hormone. This other example over here is what's called golden rice. Okay, golden rice is a genetically modified food. It's fortified with what's called beta-carotene. Okay, beta-carotene is what the body can convert into vitamin A. Okay, the World Health Organization estimates 1 to 2 million children die every year from vitamin A deficiency. And so this golden rice here on the right is a transgenic organism. It's a mix of bacteria and daffodil genes are mixed into the rice genome. And so, like I said, it's fortified with this beta-carotene that can be converted into vitamin A, but there are certain regulations in place that are actually making it so this golden rice is not available. This would be, like I said, when we talk, as we go through this unit, you need to think about the ethics you think are involved with this. Okay? Is it okay? Should the regulations be in place, or should people be allowed to eat this golden rice that we don't know what it's going to do to them when they're now eating this mix of bacteria, daffodil genes, and the rice genome? Okay, so we'll look at the other biotechnologies on the next video and be thinking some more about the ethics side of it.